MetJ repressor protein DNA complex. MetJ is a protein that represses DNA transcription by binding to DNA.
MetJ acts as a repressor of DNA transcription by binding to specific operator sequences on DNA fragments. Two dimeric repressor molecules bind to an 18 base pair DNA fragment. These molecules are 8 base pairs apart from one another. The green in the figure demonstrates the two MetJ monomers, which are bound to the DNA, which is shown in orange. Double-stranded antiparallel beta ribbons specifically bind in the major groove of B-form DNA. The R groups of lysine 23 and threonine 25 residues hydrogen bond with the DNA base pairs on the operator sequence. The yellow residue in the figure represents the threonine on the beta sheet of each subunit, and the light blue represents the lysine residues on the beta sheets.
The secondary structures for the protein consist of 3 alpha helices and 1 beta sheet. That includes the amino acid residues involved in protein DNA interactions. This is per monomer, which is shown in the figure on the right. The tertiary structure is a homodimer formation where 2 monomers come together, so the overall structure contains 6 alpha helices and 2 beta sheets that are antiparallel to one another. The quaternary structure of the protein consists of the MetJ molecule that comes together when bound to DNA, forming a left-handed superhelix. 2 MetJ complexes can come together to form a tetramer when they are bound to adjacent or tandem Met boxes. There is also a co-repressor that binds to MetJ and aids in the affinity of DNA binding to the protein. The repressor has 2 flexible loops made from residues 12 through 20 and 78 through 83. The loops are represented in red on the pymal figure to the right. When the repressor is bound to DNA, the 12 through 20 loop had completely different conformation from the beta hairpin it adapts in the holorepressor and wraps around the phosphate backbone. The glycine 15 has a positive angle in both conformations as residue 2 of the type 2 turn in the holorepressor and residue 3 of the type 2 turn in the complex, allowing it to donate a hydrogen bond to a phosphate oxygen.
For the basic function, the operator, which is the Met box sequence on DNA, is the segment where the repressor binds. The repressor here is the MetJ transcription factor. Transcription is prevented when the repressor binds to the operator, thereby limiting gene expression. For the operator conformation, DNA is in its B form. Binding of MetJ to the operator causes a sharp bend in the DNA helix at the center of the Met box. This allows DNA to be in full contact with the protein, which is MetJ, across the entire sequence. The binding causes a 23-degree angle bend towards the major groove of DNA. The bend and groove width variations are essential to the complementary fit of DNA to the repressor surfaces and to establish tight protein-protein contact.
The Met box is an 8 base pair sequence on the operator. The consensus sequence for MetJ is AGA, CG, TCT. This sequence has the highest affinity for DNA protein binding. Many different sequences can be Met boxes, but they have relatively low affinity compared to the consensus sequence. S-adenosyl methionine binds to MetJ and enhances its affinity. In natural operators, there are usually 2 to 5 tandem repeats of Met boxes. This gives increased tightness of binding, which will be further discussed later on. The figure on the right shows 2 tandem Met box sequences, and you can see that lysine can bind to guanine, thymine, or adenine, while threonine binds only to adenine. This video shows the repressor and Met box interactions, which are hydrogen bonds within the major groove. The white sequence represents a Met box that is found on the DNA. You can also see the 2 antiparallel beta sheets in green and pink. The lysine and threonine residues are specifically involved in binding.
MetJ binds to the operator sequence through hydrogen bonding between lysine 23 and threonine 25 to thymine and guanine or adenine DNA base pairs respectively. MetJ acts as a holorepressor because it inhibits gene expression and it has a corepressor, which is the SAM molecule. Upon binding to DNA, it wraps around the phosphate backbone. In the absence of the SAM molecule, MetJ can still bind relatively strongly to the consensus Met box if it is in a tetramer form. This is due to the presence of several Met boxes. Binding here is still relatively weak and not as tight as it would be with the SAM present. This protein binding is cooperative, so as the MetJ concentration increases, binding to DNA occurs more tightly and more quickly.
S-adenosylmethionine, which we refer to as SAM, acts as a corepressor with MetJ. One SAM molecule binds each MetJ monomer in a weak, non-cooperative manner. The SAM corepressor is not required in order for MetJ to bind to the DNA, but without the SAM molecule, the binding is very weak, and a single MetJ complex will not stay bound to DNA for long. SAM mediates an increase in the affinity from MetJ to DNA, but has no effect on the specificity of the interaction. Long-range electrostatic interactions occur from the binding of SAM to each MetJ complex. The sulfur in this reaction causes a positive electrostatic potential on the protein, helping it to interact with DNA. SAM molecule lies on the outside of the MetJ complex and it's distant from the DNA binding site. There is no major conformational change of MetJ upon binding to SAM.
This video shows the MetJ complex in green bound to the corepressor SAM molecules in blue. Studies have shown that MetJ binds cooperatively to the operator sites on DNA. By increasing the amount of Met boxes that are present on the operator, there is a possibility of additional binding interactions between DNA and the MetJ. In the presence of more protein complexes, the binding interactions become tighter and further binding to DNA is increasingly cooperative. These protein-protein interactions are mediated by extensive hydrophobic contact between anti-parallel alpha helices with a network of water-mediated hydrogen bonding. Hydrophobic residue interactions here predominate near the ends of the helices. Hydrogen bonds in the center of these helices link specific residues, such as threonine, glycine, and arginine, away from water, preventing non-favorable interactions.
The MetJ repressor binds to the operator on DNA, which blocks the transcriptase from binding to the DNA and preventing further transcription processes because the binding causes a conformational change and results in a 23-degree bend on the DNA alpha helix. Tetramer formation of the protein on DNA is required for repression to achieve tighter binding of the operator and the repressor. This figure shows a tetramer complex of MetJ binding to tandem Met boxes.
How MetJ structure is important for its function. The lysine and threonine residues of MetJ on the anti-parallel beta sheets of the protein interact with the Met box sequence of the operator through hydrogen bonding in the major groove of B-form DNA. This interaction causes a conformational change as the helix of DNA bends to a 23-degree angle. This gives the operator complete interaction with the protein along the entire operator sequence. MetJ binds cooperatively but weakly to the consensus Met box, with binding affinity further enhanced when the SAM corepressor is bound to the MetJ. The corepressor also allows binding to less specific Met boxes. By binding to the operator, MetJ occupies the site where a transcription enzyme would normally bind. This effectively prevents the transcription process from progressing in the presence of MetJ. This also limits gene expression. Through these specific aspects of the MetJ structure, it is able to bind to DNA and block other transcription factors, allowing it to act as a transcriptional repressor.
Now, please enjoy our MetJ mixtape. I've got my MetJ binding to DNA. 2 dimers bound 8 base pairs away. And it sure would like some SAM to help to increase its affinity today. When it's bound, that DNA is gonna bend. It's gonna bend to 23. It's gonna keep from transcripting. That DNA is gonna bend when it's bound. It's gonna bind to the major group. It's gonna bind to a dimer. It's as a co-repressor, or it can bind as a tetramer.